Hello, this is Kumar and Aditya's Science Experiment, or cooking show. Today, we will be finding out how to extract DNA from everyday fruit. This is some of the materials you need for this experiment. Salt, alcohol, you'll need your parents' permission for this, and be sure to have your parents when you're using it. Some a clear container, and some measuring tools for the clear containers. A timer, an egg timer, would be nice, but any other sort of timer. Even a clock would be suitable. Some toothpicks. This is for the end, and we'll explain that. We'll need 4 bowls, 5 if you want, and we'll also need a couple of fruits, tissue paper, a cutting board, a knife, a mixer, and a tea strainer. We'll explain the use of all these and why we need them later in the experiment.
Okay, so the first fruit that we're going to use is an orange. So we're only going to use half of it because, you don't need the whole thing. Although I can rip it in with my hands, I need a knife. And I'm going to take piece by piece, or some might call it slice by slice, and put it in the blender. Not only is this going in the blender, but 2 tablespoons of water. Cold water, that is. So, 1, 2. After that, you take some salt, the salt that you have, and take a large pinch with your hands. Take a pinch of it. And put it in the blender. Maybe some more, depending on how much you have.
Go ahead.
Okay, so here it is. Everything in the little jar. Now we just put it in the blender. And be careful this part because you do not, I repeat, do not want any of the contents spilling out because that is going to cause a major catastrophe. So, when you're ready and when you have all your strength put together on forcing the top of the lid on the container, turn it on. Well, you want it to be a nice juice. You don't want it to be, like thick and everything because that's just harder. That just makes everything harder for you. Life is hard. But it can be, you can make it easier if you try. So now what you want to do is strain out all the big, thick, chunky parts because you want to take away the cell membrane. The cell membrane is what prevents you from getting getting the DNA directly. So here we go, pouring in the strainer. Right now, what you have to do is squeeze all this thing out because you want as much juice as you can get for your DNA sample. So here I am, just pushing all the pulp away. Okay. So when it looks like it's almost done, and you have enough, just put it away, take a paper towel, just wipe your hands. So what you want to do now is take the detergent to mix it away. Although you did put some cell membrane away, and you kind of pushed it away by means of straining, you want to use the detergent to fully dissolve what is remaining. So, here we go. Slowly pour it in. After pouring it in, you just want to wait for some time. Approximately 10 minutes or so. That's when our handy dandy timer comes in play. Time's up. So, party gets started. This is what we have. I'm not going to show you directly because if I do, it's going to spill out, but if you can see, kind of, that's how it looks like. Now, the next thing we're going to do is put enzymes. Well, you can use many substances for this. For example, meat tenderizer, pineapple juice, your contact lenses if you have any, their washing solution or their solution for taking care of them. Well, we're using pineapple juice for the experiment. And you can use whatever you have, but as I said, for this experiment, we're using pineapple juice. So what I'm just going to do is pour a bit into here. Just sit and stir gently. You put this substance, into the container. You might only need half of the container full, but you won't need all of it. Here we go. Be careful. Now you have your solution in this thing, in this container. All you need to do is now take this alcohol, take approximately 5 milliliters of it, and pour it into here at an angle, which will be explained in a further step. Be sure to cap the alcohol because you do not want to spill it, for it can create a big mess and quite shortly it can stink a bit. And I've experienced that. So pour this at an angle. This is because we want the two substances to separate so we can get the DNA from the top. We don't want them to mix. And if you put it like this and pour it, it's going to mix. So in order to stop that from happening, we're going to tilt it at an angle so these two substances will not be in the same area. So here we go. Slowly though. As you can see now, there are two different areas. A thin area, and then there's DNA, and then below that, there's other substances. Now, for the extraction of the DNA. For this, make sure your tube is in a place where it will not slip, because you might need to keep it there for some time. And while you're at it, take out a toothpick or some other tool that is similar to this, which you can easily take out the DNA. It needs to be a little thin rod, kind of like the surface of a toothpick and the texture of a toothpick. So what you want to do is slowly put it in and twist it. Twist it so the DNA forms like a little cotton ball on the tip. You know how when you go to a carnival and they twist it to get some cotton candy? Similarly, that's what you want to do with the DNA. So, here we go. So, an example would be this, as you can see, for a small blob of DNA. This blob, which looks like snot, is what we humans call DNA.
Hello,